Okay, so now we begin with the development and anatomy of female sexual organs. First of all, we'll see how the embryology of this basically works. The gonadal rudiments appear as a genital ridge overlying the embryonic kidney in the intermediate mesoderm. So here you can see the mesoderm. This is the lateral bit. This is the intermediate mesoderm, which creates the pronephros. The pronephros then becomes the mesonephros, which then becomes the metanephros. The mesonephros helps to differentiate into the gonads, and the metanephros becomes the kidneys. This is the paraxial mesoderm, this is the neural tube, the notochord, and the yolk sac. Now, this happens in the fourth week of embryonic life, and they remain sexually inactive or indifferent until the seventh. So, in the seventh week, the undifferentiated gonad has the potential to become either a testis or an ovary, and hence it's termed bipotential. Okay, now let's look at this. This is the neural tube. We can see a longitudinal section here. These are the gonadal ridges that appear through the intermediate mesoderm. This is the hindgut. So this is the foregut, hindgut, and this is the heart. So you can see that they somehow overlie the embryonic kidney part. So this is the mesonephric duct, the mesonephric tubules, the nephrogenic cord, the interaction between the metanephric blastema and ureteric bud develops the entire kidney system. And these are the future gonads. This is genital ridge that somehow overlies the embryonic kidney, right? Now, why do we call it the undifferentiated gonad? Why do we call it bipotential? Because it has the potential to become either a testes or an ovary, right? And the chromosomal complement of the zygote determines whether the gonad becomes a testes or an ovary. So the development of either the testes or an ovary is an active gene-directed process. In the male, the activity of the SRY gene. So let's look at the SRY gene here. So this is a bipotential gonad, right? Now this can either become a testes or an ovary. In the male, the presence of this SRY gene, this is the sex determining region of the Y chromosome, right? So the Y chromosome is the sex determining region is the SRY gene. It causes the gonad to begin development into a testes. Okay? In the past, the ovarian development was considered a default development due to solely the absence of SRY. But in the last 10 years, ovarian determining genes have also been found that actively lead to the development of the female gonad. So you have some female specific signals now, right? But in the last 10 years, people used to think that the absence of the SRY gene leads to the ovarian development. Okay, the fetus has two set of structures called the Mullerian and the Wolffian duct. So there's a Mullerian duct and there's a Wolffian duct. Okay, the Mullerian is also known as a paramesonephric duct, and the Wolffian is also known as the mesonephric duct, which have the potential to develop into a male or female internal and external genitalia, respectively. Development of the male sexual organs as the gonad develops into a testes, it differentiates into two cell types. The Sertoli cells produce anti-Mullerian hormone, and I'll take you to the image of the Sertoli cells. Let's take a minute to understand this image. Right? Okay. So we have the testes, we have the Leydig cells, we have the Sertoli cells, right? And let's go on and see what these cells basically do. The Sertoli cells produce anti-Mullerian hormone and the Leydig cells produce testosterone. Now what happens is that the testosterone, the Sertoli cell, the anti-Mullerian hormone, what does it basically do? The anti-Mullerian hormone suppresses further development of the Mullerian ducts, whereas the testosterone stimulates the Wolffian ducts, right? So the Sertoli cells are going to suppress the Mullerian ducts and is going to cause its regression, whereas the Leydig cells are going to make testosterone, which are actually going to stimulate the Wolffian ducts to develop into the vas deferens, the epididymis, and the seminal vesicles. So the vas didymis, the epididymis, and the semiginral vesicles are formed from the Leydig cells, creating testosterone, which is going to create stimulate the Wolffian ducts, which are going to create the vas deferens, seminal vesicles, and epididymis. Right. In addition, in the external genital skin, the testosterone is converted by the enzyme 5-alpha reductase into dihydrotestosterone. This acts to virilize the external genitalia. So this dihydrotestosterone causes virilization of the external genitalia. The genital tubercle becomes the penis, and the labioscrotal folds fuse to form the scrotum. The urogenital folds fuse along the ventral surface of the penis and enclose the urethra so that it opens at the tip of the penis. Okay? So basically, the 5-alpha reductase gives you the DHT dihydrotestosterone. This acts to realize the external genitalia. And then the genital tubercle, which we saw in the beginning, becomes the penis. And the labioscrotal folds fuse to form the scrotum. The urogenital folds fuse along the ventral surface of the penis and enclose the urethra so that it opens at the tip of the penis. Now, development of the female sexual organs. Now let's look at the development of the female sexual organs. A little bit we can see here, right? So in the primitive ovary, we have granulosa cells. These are derived from the proliferating coelomic epithelium surrounding the germ cells and from primordial follicles. I'll show you another picture of the granulosa cells here. I really like this picture. So can you see this? Okay. So basically, okay. So in the primitive ovary, granulosa cells derive from the proliferating coelomic epithelium surround the germ cells and from primordial follicles. Each primordial follicle consists of an oocyte within a single layer of granulosa cells. Theca cells develop from the proliferating coelomic epithelium and are separated from the granulosa cells by a basal lamina. So we're going to see these. This is the basal lamina. These are the thecal cells. This is the primary follicle, and each primary or primordial follicle consists of an oocyte with a single layer. This is a single layer of cells, of granulosa cells, right? So this is a primary follicle that is surrounded by a single layer of granulosa cells. Now these cells develop from the proliferating coelomic epithelium and are separated from the granulosa cells by a basal lamina. So now we see the basal lamina here. We see all these granulosa cells, right? The maximum number of primordial follicles is reached at 20 weeks gestation, where there are 6 to 7 million primordial follicles present. This is important because this is an exam question. So I'm just gonna write it down here. So the maximum number of primordial follicles is reached at 20 weeks gestation, where there are 6 to 7 million primordial follicles present. Now, what happens is these numbers, the numbers of these reduce by atresia, and at birth only 1 to 2 million remain. So at birth only 1 to 2 million remain, and these numbers reduce by atresia. The numbers of these reduce, and then atresia continues throughout life, and by menarche only 300,000 to 400,000 are present, and by menopause none are present. Okay, so these numbers continue to reduce through atresia, and I'm going to write this down here. This is also important. So the numbers of these reduce by atresia, and birth only 1 to 2 million remain. Atresia continues throughout life, and by menarche only 300,000 to 400,000 are present, and by menopause none are present. The development of an oocyte within a primordial follicle is arrested at the prophase of its first meiotic division. This is also very important. The development of an oocyte within a primordial follicle is arrested at the prophase of its first meiotic division. It remains in that state until it undergoes atresia or enters the meiotic process preceding ovulation. In the female, the absence of testicular anti-Mullerian hormone allows the Mullerian structures to develop, and the female reproductive tract develops from these paired ducts. So I'll show you these paired ducts. So these are the two ducts, and the absence of the anti-Mullerian hormone and some ovarian genes leads to these two Mullerian ducts. The ducts fuse, and the fused Mullerian duct, then these are the two primordial ovaries. They don't fuse, neither do the fallopian tubes. And what happens is the absence of the testicular anti-Mullerian hormone allows the Mullerian structures to develop, and the female reproductive tract develops from these paired ducts. The proximal two-thirds of the vagina develop from the paired Mullerian ducts, which grow in a caudal and medial direction and fuse in the midline. The midline fusion of these structures produces the uterus. Okay, the midline fusion of these structures produce the uterus, also the cervix and the upper vagina. So this area is the uterus, the cervix, and the upper vagina, and the unfused caudal segments form the fallopian tubes. These unfused caudal sections form the two fallopian tubes. The cells proliferate from the upper portion of the urogenital sinus to form structures called the sino-vaginal bulbs. I'm going to show you the sino-vaginal bulbs. There was definitely a picture of the sino-vaginal bulb somewhere here. Here. This is the sino-vaginal bulb. Can you see this here? This is the sino-vaginal bulb. This is the caudal tip of the Mullerian tube, and this is the sino-vaginal bulb. It also is known as the vaginal plate. So what did we just say? The midline fusion of these structures produces the uterus, cervix, and upper vagina, and the unfused caudal segments form the fallopian tubes. Now, there's something, the cells proliferate from the upper portion of the urogenital sinus. This is the urogenital sinus. The cells from the upper portion of the urogenital sinus proliferate to form structures called the sino-vaginal bulbs. This is the sino-vaginal bulb. The caudal extension of the Mullerian duct, this caudal tip of the Mullerian duct, the caudal extension of the Mullerian ducts projects into the posterior wall of the urogenital sinus as the Mullerian tubercle. So this is where the Mullerian tubercle is. The caudal part of these Mullerian ducts come here, and they form the Mullerian tubercle, right? The Mullerian tubercles and the urogenital sinus both fuse to form vaginal plates. This is the vaginal plate, which extend from the Mullerian ducts to the urogenital sinus. This plate begins to canalize. When I said something canalizes, it starts to become hollow, starting at the hymen and proceeding upwards to the cervix in the sixth embryonic month. So in the sixth embryonic month, till the sixth embryonic month, this canal begins to canalize, right? Now, external female genitalia. The external genitalia do not virilize in the absence of testosterone, because the fifth and seventh weeks of life, the cloacal folds, which are a pair of swellings adjacent to the cloacal membrane. Let me show you the cloacal membrane and cloacal folds. Here, this is the cloaca. Okay. So what happens is that the cloacal folds, which are a pair of swellings adjacent to the cloacal membrane, fuse anteriorly to become the genital tubercle. There's also a picture of the genital tubercle here. I will show you that. Okay, I can't seem to find the genital tubercle here. It's lost, I guess. So this gonadal ridge ultimately becomes genital tubercle. Or let me find a picture of it.